whereas these cells, the pyramidal cells, are large and project long distances, and they excite the cells that they target. If we could figure out the role that each of these cell types plays in the brain, we could understand how they work together to create thoughts, emotions, and behaviors, and how flaws in specific cell types may lead to devastating brain disorders like schizophrenia, Parkinson's, and depression. Now, MIT researchers are working to develop tools that allow us to do exactly that. It is now possible to precisely control the activity of specific neurons, and therefore understand how each neuron contributes to brain function. Remarkably, these high-tech tools have come from microorganisms, such as this pond algae called Chlamydomonas. Chlamydomonas needs light for photosynthesis, and it swims toward light with the help of a small organelle called an eye spot. The eye spot contains a light-sensitive protein called channel rhodopsin, which responds to blue light by moving positively charged ions across the eye spot membrane, causing the voltage of the eye spot to change, a bit like a battery being charged by a solar cell. But neurons are also electrical devices, and their voltage would also change if channel rhodopsin could be inserted into their membranes and then illuminated. So, researchers took a piece of DNA that encodes for the channel rhodopsin protein. Then, they put it into neurons, using a safe and effective method for delivering a gene, the same kind being used in human gene therapy trials. The neurons expressed the DNA, manufacturing copies of the channel rhodopsin protein and installing them in their membranes. As predicted, the researchers were now able to activate these neurons using light, without affecting any of the neighboring neurons. This technique, called optogenetics, allows researchers to drive the activity of specific neuron types to see how they affect brain activity and behavior.